Okay. So good evening to most of you here, and thanks for joining us. So kind of our goal today is to talk about cell counting. Not really something that I think a lot of people, and I'm guilty of this myself when I was a graduate student. Cell counting is something that I would kind of rush through. You wanted to finish it up and get on with the next steps of your experiment. But really, when you think about it, if you're doing a cell-based workflow, cell-based experiments, you have two inputs. You have whatever your treatment is. So, that might be a drug or some kind of virus titer or something like that. And then you have your cells. So if your cell counts, your input into your experiment from the cell count isn't accurate or at least consistent, then everything downstream of that, all of your experiments that you do after that cell count are also not going to be accurate or consistent. So kind of the goal here is to go over just 7 tips, things that you can keep in mind while you're doing a cell count that will, it's not going to slow you down significantly, but it's going to make sure that you're getting consistent inputs into your cell count when you're doing your cell count, so that you have accurate numbers going into the next step of your experiment. So the flow here, we're going to do just a quick background on cell counting. That should only take a few minutes, and then we'll go into the 7 tips for improved cell counting. And then at the end, I actually have a cell drop where I'm in the lab here at DeNovix. So I have a cell drop and some live cell samples with me. So if there's any questions, if there's anything in particular that someone wants to see, I can do that. We can do some experiments or whatever here in the lab. So that is the plan. All right, so sorry, I'm just moving where the there we go. So 2 basic methods of cell counting. The first is your flow-based. So this is flow cytometers and Coulter counters. These types of cell counters are great for getting lots of data, really fast counts of large numbers of cells, and comparing populations within those sets of cells. They're not really the best. I think a lot of people, this was surprising to me, and a lot of people don't know this, they're not really the best for giving you accurate cells per mil counts. They recommend that you either run a bead standard if you want an accurate cell per mil count with your run, or you need to make sure that you're regularly calibrating the pump on your flow-based cell counter. And also, I mean, if you just need a cell per mil count, a flow cytometer is going to be really expensive. It's going to take a lot of time just to get a run for a really simple number that you can get a lot easier using our second method, which would be the image-based cell count, or either a manual hemocytometer or an automated hemocytometer. These are much lower throughput, of course, but if you're looking to get an accurate cell per mil count for going into the next step of an experiment, seeding a plate, seeding a flask, something like that, this really is going to be your quickest and most accurate way to do it. So just quick overview of how a hemocytometer works. Really all you're doing is you're counting the number of cells in a fixed volume. So in a manual hemocytometer, it's 1 millimeter by 1 millimeter, and then that has a cover slip on top of it that gives you 100 micron chamber height, and you end up doing a quick calculation on that to get a cell per mil count. Only reason I mention that is just because I think a lot of people don't realize that automated hemocytometers work using the exact same principle. You're just looking at the number of cells in a fixed volume. There's a few key differences, of course. One, you have a much larger field of view. With the cell drop, it's about 3 times larger than what you'd get with a manual hemocytometer. Doing the actual count is much faster, so it makes it possible to do multiple replicates pretty easily, which of course is giving you more data, more representative look at your sample, which could increase your accuracy. And also using automated hemocytometers with pre-programmed algorithms to count is going to be more consistent than if you have a person who, day to day might slightly vary how they count. Say it's Friday evening, like it is for you folks there. They might maybe be looking to get through the count pretty quickly so they can finish seating their plate and get home. With an automated hemocytometer, you don't have that kind of an issue because the count is going to be consistent every single time. And then at the bottom of this page here, I just have some data that we have from a published TechNote showing that the consistency on an automated cell counter is about 3 times better than what we got with a manual hemocytometer. Okay, so that's the quick overview of cell counting. We'll get into the 7 tips to make sure that your cell counts are accurate now. Those 7 tips are to clean the sample surface, mix well before loading, minimize cell clumping, choose bright field or fluorescence for your counts, optimize the instrument settings if you're using an automated hemocytometer or a flow cytometer or something like that, use an appropriate chamber height, and adjust the count parameters. So number 1, cleaning the sample surfaces. Seems pretty basic. With a manual hemocytometer, you want to wipe away whatever sample you had on there with a Kim wipe, apply some ethanol to the surfaces and wipe again to make sure it's clean. And then the hemocytometer manufacturers recommend that you store it in a case with a cover slip on top of it to prevent dust from getting on there. With a disposable slide, you want to throw that slide away. Something that we've actually gotten questions about, believe it or not, is people want to pull apart the glued plastic on these disposable slides and then try to clean and reuse the slides. Please do not do that. That's not good technique, and you're not going to get an accurate count from that slide the second time. With a cell drop, so the automated hemocytometer with reusable surfaces, it's pretty much exactly the same as the manual hemocytometer. You just take a Kim wipe, you wipe the surfaces clean. After your final sample, say do 4 or 5 samples of a cell, then you just would want to clean it with ethanol, like the manual hemocytometer before the next run. So occasionally, or after you clean it or between runs, you might see there's a little bit of dust or something like that that gets left on the surface. It's not anything you need to be worried about, because when you load that next sample, as you'll see here in this video, that should just get flushed away and it won't be a problem for the count. So if you do see that on your manual hemocytometer or on your cell drop, not something to be concerned about. You don't have to clean it over and over again to get it perfectly clean. The only time that you might want to do some additional cleaning is if you notice that the previous sample has dried up and caked onto the surface. Then you might want to clean it additionally with ethanol a couple extra times. All right, so tip number 2, make sure that you mix the sample well before loading. I think we all understand that cells are not in solution, they're in suspension, and that if you let that tube sit there after mixing it for a few minutes, that the cells are going to all settle to the bottom of the tube. What wasn't intuitive to me is how quickly that actually can happen and how much it can affect the counts. if you don't use good technique here. So to prove that, we did a quick experiment. We just took some styrofoam beads and we got them to a target concentration of 1.5 million beads per mil. And then we took that tube of 1.5 million beads per mil and we mixed it well just by taking a pipette, pipetting up and down a bunch of times, and then loaded that sample immediately onto a cell counter. Then we took that same tube, mixed it well, and then just waited 15 seconds. So kind of trying to mimic the amount of time that if you got briefly distracted by a text message or somebody else coming into the lab and asking you a quick yes or no question, the amount of time that might take. And then we took the sample from either near the top or near the bottom of that same tube. And what we saw with the well-mixed sample, here's just an example image, is that it came in about on target, 1.6 million cells per mil. That exact same sample where we took the sample from near the bottom, like not all the way at the bottom, but like the bottom quarter of the tube, it came out at 2.5 million cells per mil. So already 1 million more cells per mil than the target, and that's just after 15 seconds. And then when we took that sample from the top of the tube after 15 seconds, it was 600,000 cells per mil. So when we actually put that data together, there was a 5 times difference in the concentration between the bottom of the tube and the top of the tube after only 15 seconds of letting those beads settle in the tube versus the correct answer of 1.5 million. So very important to make sure that you mix and then load immediately if you want to get accurate results from any kind of cell counter. Tip number 3 here is to minimize cell clumping. So when we're talking about cell clumping, really what we're talking about is rushing through the trypsinization step. So instead of letting the trypsin digest your sample well, putting it in the incubator at 37 degrees, not waiting long enough and cells clumping up when you go to take them out and put them in your tube to move on to your next step in your experiment. So to get an accurate cell count, you need a good representation of the total sample that's in that tube. And clumps will misrepresent the sample because if you have a big clump, it throws off the numbers of that whole tube. And they're also, if you're using a manual counting method, they're just hard to count. So, I mean, if you look here at this poorly digested sample, if I was manually trying to count this, I'm probably not going to get a very accurate count of these clumps that are in the bottom right of this image here. The other thing is if you look at the left of this image versus the right of this image, there's going to be probably close to like a 5x difference in the count here versus looking at a properly digested sample. Everything's easy to count either manually or and everything is evenly distributed across the screen. So just to kind of get an idea of samples, we did another quick experiment. We took CHO cells, seeded 2 flasks, grew them for the exact same amount of time, and then one we poorly digested, so kind of rushed through the digestion step, and the other one we digested according to the manufacturer's protocol, the trypsin manufacturer's protocol. And we took 5 replicates of each the poorly digested sample and the properly digested sample. On average, there wasn't that much of a difference, and which is good. That's what I would expect. But the standard deviation was 10 times higher for your poorly digested sample. So if we didn't do 5 replicates, there's a good chance we would have gotten a fairly fairly inaccurate count from just one of those replicates. And then together they kind of all offset each other and averaged out. I don't know anybody that takes 5 replicates to get an accurate cell count. Everybody just wants to take one and move on. So making sure that you're properly digesting your cells and not having clumps in that sample before you count it is very important. And it's also just very important for later when you go to seed your plate or your flasks to get an even number of cells in that plate between the wells. So tip number 4, making sure that you choose the correct technique to count your live dead samples. So this means choosing between a bright field based technique or a fluorescence based technique. Depending on whether you're using primary cells or just a standard tissue culture cell, this could be a very important choice in making sure that you get accurate data. If you're working mostly with primary cells, so whole blood or ground up lymph nodes or something like that, for example, you're going to have a lot of additional debris, red blood cells, tissue debris in that sample that are going to be tough to count with a colorimetric stain or trypan blue. If you use fluorescence for that sample, that's going to just target the cells of interest in that particular sample. So almost all the time people are looking for nucleated cells, white blood cells or something like that in this type of a sample. So a fluorescence stain, Aopi, is going to selectively pick just the nucleated cells and exclude the debris from that sample. So just to go into what Aopi is real quick, because I think everybody knows what trypan is, but not everybody is familiar with these dyes. Aopi is a combination of 2 fluorescent dyes, acridine orange and prapidium iodide. Acridine orange is a live cell stain. It passes through the membrane of all cells and stains the nucleic acids in those cells green, or they fluoresce green. PI cannot enter into live cells. It can only pass through compromised cell membranes, so cells that are dead or dying, and then it stains the nucleic acids in those cells red. AO enters both live and dead cells, but the PI dye quenches the green signal from AO. So when you look at it under a microscope, cells that have been stained with this combination of dyes, if there's a green cell, it must be a live cell, and a red cell must be a dead cell. So here's an example of a primary cell. This is a whole blood sample that had an isolation done, just looking for the nucleated cells. But as you can see, there's a lot of things not being counted in this image. So those could be red blood cells, they could be platelets, just could be other tissue debris from the isolation. So counting with fluorescence, you can see it's very clear what is and what isn't a cell. The cells are bright green, lighting up, easy to count. And the dead cells are bright red, lighting up and easy to count. Taking that exact same sample and trying to count it with trypan blue stain, things become a lot more subjective. Deciding what is and what isn't a cell is kind of guessing work at this point, because red blood cells can look exactly like live cells. And you can see here in this image that it looks like it's being overcounted. So we have some data, we have some published data on this, and there's other places that this data has been published as well in the literature. This is kind of a well-known phenomenon that trypan blue overcounts primary cell samples. We found that it can be by a factor of up to 2 times when compared to an Aopi sample for primary cell samples. And using the same dyes, Aopi, when you take a nice clean tissue culture sample, something that doesn't have debris from red blood cells or tissues in it, you get very good agreement between Aopi and trypan blue using a clean tissue culture sample. Okay, so tip number 5 is to make sure that you properly optimize the instrument settings. When we're talking about instrument settings, we're talking about things like focus and exposure. Um, Trypan blue, again, is more difficult, it's more subjective, and it's very vulnerable to if the sample is out of focus or if the exposure is not quite right, because that can make live cells appear to be dead or dead cells appear to be live in the count. Fluorescent techniques are much more forgiving, but I have some examples here where you can still, by not having the focus set correctly, really screw up the count by in fluorescent techniques by not having the focus or exposure set correctly. So as I was saying, the fluorescent techniques are more forgiving, but still important to get these instrument settings right. So here's an example of a sample, trypan blue stain sample that's well focused. You have dark outlines around the live cells and a bright white center in those cells. It looks like you've got about 90% viability with this count, and your live cells per mil is about 850,000. That exact same sample, just kind of adjusting the focus poorly, you've now gone down to about 60% viability, and your live cells per mil have gone down to about 600,000. So just by adjusting the focus, I've really managed to change the final result here. And then also with exposure, here's again a trypan sample, properly exposed. So you can see there's a nice gray background, and the centers of the live cells are bright white against that that gray background. 80 or so % viability and about 600,000 live cells per mil. When I have this same sample overexposed, you can see it really washes out the image. You don't have a good background to count for the algorithm to count well against. And your viability has now gone up to about 92%. And also, I just want to point out the reason I chose this particular example. If you look at here at this cell at the top and the bottom, there's those two dark cells. In the properly exposed example here, it's very clear that those are not cells. Those are bubbles that have somehow gotten into the sample. If you've washed out your image because the exposure is too high, now to me, those look just like dead cells. And if I were manually counting this, I would probably count those as dead cells. And you could hide all kinds of other things with an exposure that's too high, and that washes out the image. You might not see contamination in this sample, or you could have bacteria contamination or fungus contamination in this sample, and you just wouldn't see it because it's too bright to see any of those details in the image. Moving on to fluorescence, here's a properly focused fluorescent example. This is in bright field, but it also properly focused in the green channel. And you get a count with about 90% viability and about 850,000 live cells per mil. That same sample, poorly focused, here the bright field channel, you can see that it's kind of out of focus. The centers don't look as sharp. And the green signal gets very dim in this situation. And what happens when that green signal gets dimmer is that those cells don't get counted properly. Now you're getting a lot more dead cells being counted where previously they were being counted as live cells. So your viability has gone from over 90% now down to under 80%. Sorry, so very important both, especially for trypan blue, but in both techniques to make sure, just take a look real quick at the focus and the exposure. Don't trust that it's been set correctly by the previous person, and adjust if needed to get the most accurate counts. All right, tip number 6 here, make sure that you're using the appropriate dilution or adjusting the chamber height before your count. So this is just talking about making sure that you have enough cells to get a representative count of what's actually in that tube. Hemocytometer manufacturers actually recommend that you have between 100 and 200 cells per square. So, there's 8 squares on that, the full hemocytometer Neubauer grid. They recommend 100 cells per square. That, I don't know anybody that actually adheres to that particular suggestion. I think most people, in fact, most of the time it falls outside of that. But I think the point is to make sure that you're getting a good representative look at your sample. If you're using the cell drop, we have a unique feature where we can actually move the chamber height. So we can raise or lower the height of our counting chamber depending on the concentration of your cells. So if you have too few cells or too many cells, like I'm showing here in this example, where you either have cells that are way too dense that would be impossible to count, and you can't even see background, so it's really impossible to know what's cell and what's not, or you have too sparse of cells where you only have, it looks like 5 live cells in this image here on the right, you can raise or lower the chamber height and then load more or less sample. So essentially you're doing either a dilution or a concentration of the cells to get a more accurate count of that sample. So when we lower those dense cells, we lower the chamber height down to 50 microns. Essentially, we've cut the number of cells in the field of view in half, and now you can see some background and potentially get a good count with an automated counter in that image. For the sparse cells, we've raised the chamber height to 400 microns. You've loaded 4 times as much sample, so now you have more cells in that field of view to get a more accurate count of what's actually in that tube. Before, we only had 5 cells in that field of view. If one of those cells had just sort of floated off the bottom of the chamber and gone out of the field of view, then you've changed your count by 20%. Now at 400 microns, looks like we have 35, 40 or something like that cells in that image, and losing 1 or 2 cells isn't going to significantly change what your count is. Okay, so last tip that we have is to adjust the count parameters. This one applies only to automated cell counters. Automated cell counters have image recognition software that in general are meant to be starting points to get a good count. For a lot of cell lines, they'll work very well, but each cell line has some slight differences to it. And we're trusting that our users are going to use the parameters and the tools that they have at their disposal to adjust those and get an accurate count for their particular cell line. So you're going to look at the image that you get and verify that it's counting correctly for you. The cell drop uses 3 main parameters. First one is pretty simple. It's just the size gate, min-max diameter. The second one is what we call roundness, and the third is an intensity threshold. And I'll go into all of those in a little bit more detail here. Min-max diameter, pretty straightforward. You're just setting a size gate. I just have a video here showing how you can do it on a cell drop. We have a histogram that you can use that shows the cell number by diameter. So if you happen to know, that your cells are only between, 8 and 14 microns, you can go into that histogram diameter, and if you see something else in there, you can bring the gate in to make sure that it's not getting counted. You also can, in real time here, this is demonstrating, adjust the count and look at the image and see what is and what is getting counted. So that's a pretty useful tool. Zoom in on the image, see things that are cells or debris, and verify that what is a cell is getting counted by size. The next parameter is the threshold. So this is just saying how dark something needs to be to get counted as a dead cell if you're using trypan blue, or how bright something needs to be to get counted as a cell if you're using fluorescence. So our default protocol for trypan blue, the threshold is 35. So 1 would be the lightest and 99 would be the darkest. And just using our default protocol here and counting this cell sample, we got about 250,000 cells per mil and a viability of about 50%. If I change that threshold and made the threshold to get counted as a dead cell, it needs to be very dark, almost black, so I set it up to 80. You can really see how that changes the count. So now nothing is just about it as being counted as a dead cell. Your viability is now 98%, and your total cells per mil is about 125,000. So that got cut in half. Going the other way, if you bring that threshold way down, so that very light things can now get counted as a dead cell, your total cells per mil goes way up and your viability goes way down. So generally, most people are going to set these thresholds somewhere in the middle. I kind of just, in this example, set it to the extremes just to show you guys how it works. But, you know, you do have a lot of control here over what is and what isn't getting counted as a cell. That was kind of our goal, is to give our users the control if they wanted to do this. The last parameter I want to talk about is what we call roundness. This one is not quite as straightforward as the other two. So roundness is a measure of how complete the outline around something needs to be to get counted as a cell, and how geometrically circular it is to get counted as a cell. So if you set this roundness to be very high, like I've done here, so I set it to be 90. In this second example here, things need to be almost perfectly round and completely outlined to get counted as cells. And you can see doing that for the live roundness, how that changed the viability and the cells per mil compared to the original example. And then again, setting these to be very loose, everything can get counted as a cell. And again, it more than doubles your cells per mil, and it sets your viability up to almost 100% doing that. So with these parameters, it's important if you're using an automated cell counter to review the parameters for that cell counter. Some have more than others. With the cell drop, we tried to strike a nice balance, not to have too many parameters, that it gets confusing, but to have the ability to give our users a lot of control over what gets counted as a cell or not. Once you have those parameters set for a particular cell line, you don't need to go back and change them every single time. They should be sort of set it once, get it right, set it, and then it should be good after that. You might want to check the images, you know, just do a quick check of your images occasionally to make sure that something hasn't changed. You know, some tech in your lab hasn't gone and edited the parameters without you knowing it. But not something you need to worry about regularly. And at DeNovix here, we're very happy to help people. You can send us raw images and we can give you suggested count parameters back for that particular cell line. Okay, so just to summarize those 7 tips, proper technique and good understanding of parameters of your particular technique instrument is key to getting accurate cell counts. If you don't get a proper cell count, this can lead to downstream issues in your experiment. You can have inconclusive or conflicting data on your endpoint experiments, you know, like flow cytometry experiments or drug development experiments. So making sure that you get good cell inputs is very important to getting consistent research experiments done. Okay, if anybody has any questions or wants to see anything demonstrated on cell drop, we can do that now.
Yes, there's a couple of people asked if you could just run through the samples on the cell drop, that'd be cool.
Okay. Okay. Do you see that?
Yep, that's coming through. Great. Even better.
Okay. So here's our cell drop FL. Startup process, and then I have some jerkat cells here that I can do some quick measurements with. So just going to make sure they're well mixed, as we just discussed here.
So Dan, we had a question about whether the counting methods can also apply to white blood cells and red blood cells, counting by hemocytometer too. I think.
Can it apply to counting white blood cells versus red blood cells?
So I think the question is where. Burla, I think the name is. Sounds like it's, do these principles, I guess, that you've discussed apply to counting by hemocytometer too?
Yeah, that was exactly. For the most part, I mean, excluding the last one that I went over, which was, you know, the different settings, parameters on an automated cell counter. The rest of the tips that we discussed here would all apply to counting with the manual hemocytometer.
Yeah.
Okay, so just getting the focus set correctly here, and count. So what's nice about the cell drop in particular is that we give this nice big image. So as I was saying, you know, you want to check your cells, make sure that they're counting properly. Don't just trust the device. So that's kind of a real advantage that the cell drop has over some other methods of cell counting or other cell counters, that you get a real nice, clear, high quality microscope image where you can see debris or contamination in that sample. And you can also take a look and, you know, just verify that things are getting counted correctly. So, you know, as you see here, the cells are being counted. We outlined, the cells that were counted are getting outlined. You can turn that on and off, verify. You have your results here to the side. So total cell count, mean diameter, cells per mil. This is just a bright field sample, so there's no live dead count in this example here. But what's, you know, the main advantage of the cell drop over automated cell counters is, now that I'm done with this sample, all I need to do is wipe it away. Instead of, you know, having to use a plastic slide that costs me money every time and I have to throw it out, just wipe it away, and now you're ready for the next sample. See another question here. Dealing with baculovirus, can the occlusion bodies be assessed by this method? I don't think that the occlusion bodies are going to be visible on an automated cell counter. You probably would need a slightly higher end microscope for that. That said, we do have our custom methods app here that kind of gives our users a lot more freedom over how to set up parameters. Our other apps that we have, like Trypan Blue, Aopi, are fairly locked. It's just set up to use those particular dyes. The custom methods really gives our users a lot of freedom to try and do things like that, look at viruses within a cell. So if the cells are big enough and the viruses are big enough, or the occlusion bodies are big enough within the cell, you might be able to set the count parameters to count occlusion bodies within that cell. I don't know anyone who has done that before, so I can't say for sure. But it could be possible, and I think it would be worth trying in a demo. Next,
some more questions come in there.
Yep. How do you differentiate between live and dead cells? So there's a couple of different ways to do it. I can just do a quick sample with Trypan Blue here, since that's one that a lot of people are familiar with. To demonstrate. And then we can do fluorescence as well. So I can show you the difference between the two. Okay, so I'm loading the Trypan Blue sample, setting proper exposure, as we talked about, and focus. Generally with the cell drop, I'm just gonna get my camera back. With the cell drop, you don't really need to focus it all that often because you have fixed surfaces. Unlike with using a plastic slide, once you get the focus plane set correctly one time, it should be consistent from then on. So, I mean, I'm making sure that the focus is correct here, but just want to point out this isn't something that you would need to. Once you get it set properly, you want to check it, but it's not something that you're going to have to readjust every single time. So I'm just going to do a count now of this Trypan Blue sample. And you can see the live cells have bright centers, and the dead cells look like dark discs. And then you have your live dead cells per mil, percent viability, all that data there is provided. I'll do a quick fluorescent sample. Well, we'll do cleaning real quick. All right, so these are the same cells, now fluorescently stained. You can see the green and the red stains that we were discussing. Not a lot of red here, not surprising since these are tissue culture cells that are pretty healthy. Just gonna adjust the exposure a bit. Focus. And count. Of course, the fluorescent count takes a little bit longer because you have to do multiple channels instead of just a single bright field channel. But it's just the results of this are just very, very clear. You can see bright green live cells versus some red dead cells, and it's just nice because there's nothing to interpret. There's no like, what do I think is a live cell versus what do you think is a dead cell? You're using chemistry with the biology to your advantage here to get a more accurate count.
If you want to look at the differences in those a little bit more, we do have some tech notes on our website that would be useful. So if you go to DeNovix.com, we have some nice tech notes on the comparisons between those different methods.
Yeah. We have a question about mean fluorescence intensity. We do have that in our GFP app here. So let me demonstrate this here. So we have an intensity histogram available here, and you can see number of cells at each intensity. So that's available in the GFP app. I think we've discussed making that available in the custom app as well. So yes, we do have the ability to do that and give you that data for your cells. What else do we have? We're working on a cancer cell line. It's very useful. How much does it cost? Demonstrate, differentiate between live and dead.
I think Rahul will pick up all the questions on pricing separately with you.
Yep.
Yes. I will talk to them and give them the best prices.
Thanks. So there are a few options available, so we need to just discuss exactly what would be appropriate for you.
It looks like we have a question about cells that are chemiluminescent. It'll still be easier to differentiate live cells from the dead. So yes, it would be. I've worked with some people who are doing cells with GFP in them. And that's just a matter of setting your fluorescence thresholds correctly. So, you want to find the background level of fluorescence or chemiluminescence that you have in your cells. And we actually have a tech note about how to do that with GFP. And then you can, from there, anything brighter than that would be counted as a live or a dead cell. Okay.
Hello, Dan.
Yep.
Hello.
Yes.
Can we use for semen analysis? Can we use this for semen analysis?
I'm sorry, I couldn't quite.
For semen, was it?
Yeah, semen analysis.
Semen analysis.
Number.
I don't think we have like a tech note or anything in particular about that, but I don't see why it wouldn't work. It'd just be something that you just would have to try out. You probably, since semen aren't terribly round, you might need to adjust the count parameters quite a bit, but it is possible.
Yeah, and to fix them clearly.
Yeah. Yes, yeah, good point. I mean, they can't be live, then they'd be moving, but. Any other questions?
Just having a quick look, see if we missed anything. I think we've got to all the questions, but please let us know if we haven't. Here you go. This is a good question here, really good question.
Yeah, that's a good question.
Yeah. So the question is, can we use a disposable slide in this for contagious samples? Can we show how?
Yeah, let me demonstrate this. Let me go back into screen share here. So we do have the ability to use these slides. There's a couple different types of slide that are compatible with the cell drop. So there's both reusable slides and disposable slides. Like for this particular question, someone's working with a highly contagious sample that they don't really want to touch. You have two options there. First, the cell drop is designed actually to go into a hood. So it can be contained within a hood. The dimensions were designed specifically to allow for that. But also you have the ability to, you have an actual groove here in the sample surface itself, or in the sample chamber itself, where a slide will fit. So if you have the correct slide, a particular, couple particular manufacturers that we can use, that would fit in there. You would set it into, we have a slide mode. So you have here in the protocol, you can do either direct pipette, sorry. You can do direct pipette or slide. So you would set that to slide mode, change the protocol. The arm moves up, and you can put your slide in there and do a count of cells with that slide. So get some cells in there to demonstrate. So pretty easy. And yeah, we have gotten that question before. So people worried about, you know, BSL level 3, BSL level 4 labs working with highly contagious samples. That's why we have that feature available. Looks like a little bit more optics of the instrument. So it has a 4x objective. It has a scientific grade CCD camera. So unlike some other cell counters, which tend to use very, let's say, cheap optics, we really wanted to have this cell counter be a high quality microscope that's also a cell counter. We want you to be able to see your cell's morphology. We want you to be able to see contamination. So, that's the first part. The specs are actually on the website. I think it's a 5 megapixel scientific grade camera. The excitation wavelengths, we just have one excitation source. It's 470, I believe, plus or minus 25. And then the emission wavelengths, 525 plus or minus 25, and 640 plus or minus 35. So that's the excitation emission. It works well for, fluorescein, acridine orange, GFP, propydium iodide. So, a lot of different dyes are covered in those excitation and emission spectra. And
those excitation wavelengths, those are LEDs, so they're kind of like super long life.
Yeah. Yes, that's a good point. These aren't lasers that are going to need to be replaced regularly or calibrated regularly or anything like that. Okay. Well, thanks so much. There was a lot of good questions that you guys had. And if anything else comes up, send an email to Rahul, and he'll forward it to either me or Andy, and we'll get back to you as soon as we can.
Yeah. Good evening, friends. I would just like to thank you for attending this webinar on 7 tips how to improve your cell counting accuracy. I'm also thankful to Andrew and Dan for such an enlightening and elaborating more on good practices to improve our counting. In short, in such a short time, Team Lab India and Team DeNovix has arranged for such a program or webinar. I would like to thank them wholeheartedly. Thank you all. And if any queries is there, I have given my email IDs. You can always write to me. And good evening to all, and have a nice evening.
All right.
Thank you, Rahul.
Good night, everybody.
Thanks
so much.